In a world where every third individual you meet could be silently battling the discomfort of irritable bowel syndrome, or IBS as it's commonly known, the need for personalized treatment strategies is more pressing than ever before. This complex disorder, presenting a kaleidoscope of symptoms, has long baffled the medical community. But today, we're here to delve into a riveting journey of discovery that unravels the mysteries of the gut microbiome in relation to IBS. Our story begins with a meticulous study involving nearly 2,000 individuals, half diagnosed with IBS and the other half serving as controls. The researchers embarked on an ambitious quest to map the microbial community within these individuals using a technique called 16S ribosomal RNA gene sequencing. The goal? To identify distinct microbial signatures associated with the different subtypes of IBS. The journey was not without obstacles. The IBS landscape is a varied one, with some individuals leaning towards diarrhea-predominant symptoms, others experiencing constipation-dominated issues, and yet others with an unclassified set of symptoms. Yet the team persevered, and what they found was nothing short of groundbreaking. In those with diarrhea-predominant and unclassified symptoms, there was a notable reduction in microbial diversity compared to the control group. Interestingly, this was not the case for those with constipation-predominant symptoms. Moreover, certain bacterial genera such as Subdoligranulum, Doria, and Eubacterium hallei were found to be abundant in diarrhea-predominant individuals, while being reduced in those with constipation-predominant symptoms. Pushing the boundaries of this investigation, the researchers also delved into the metabolic pathways. They discovered enhanced hydrogen sulfide production in diarrhea-predominant individuals, potentially contributing to their symptoms, and increased palmitoylate biosynthesis in those with constipation-predominant symptoms, which could affect stool consistency. However, the story doesn't end there. The plot thickens when depression enters the equation. IBS patients with concomitant depression exhibited significant alterations in their microbiota composition. This included a reduction in beneficial bacteria and an increase in potentially harmful ones, hinting at the intricate interplay between our gut and our brain. So, where does this leave us? This study serves as a beacon, illuminating the critical role of the microbiome in differentiating between IBS subtypes and its potential impact on treatment strategies. It highlights the need for a more integrated, personalized approach to managing IBS, one that considers diet, microbiome modulation, and mental health support. Looking forward, future research could venture into longitudinal studies to understand the long-term implications of microbiome alterations on the clinical trajectory of IBS. Such discoveries could pave the way for novel therapeutic interventions, offering hope to millions of IBS patients worldwide. This journey through the gut microbiome has offered us rich insights into the complex world of IBS. It underscores the immense potential that lies in understanding our internal microbial landscapes, nudging us closer to the day when personalized medicine becomes a reality for all those battling this multifaceted disorder. This comprehensive study has successfully identified 101 distinct gut microbiome signatures, each associated with specific subtypes of irritable bowel syndrome, IBS, and concurrent depression. This illustrates the intricate interplay between microbial populations and mental health. The researchers examined how various factors like age, sex, and diet influence the gut microbiome in IBS patients, thereby contributing to our understanding of the complex and multifactorial nature of this condition. The research delineated particular bacteria linked to the clinical features of IBS. Notably, Ruminococcus narvis, which prompts inflammatory cytokine, TNFA, production, showed higher prevalence in patients suffering from IBS-D, diarrhea-predominant IBS. In contrast, Oscillospira, associated with constipation symptoms, was more common in IBS-C, constipation-predominant IBS. Beyond these subtype-specific differences, common microbial markers such as Sotorella were identified across all subtypes, suggesting potential targets for developing universal IBS therapies. Additionally, specific bacteria like Akkermansia were recognized for their potential as probiotics, particularly beneficial for individuals adhering to lactose-free diets. A fundamental discovery of this study was the increased activity of the palmitoleic biosynthesis pathway in IBS-C, which leads to the production of palmitic acid. This acid combines with calcium to form insoluble calcium palmitate, which has been linked to increased stool hardness, a potential factor in the prevalence of constipation among IBS-C patients. The relationship between IBS and depression was explored in depth. The findings indicated that IBS patients with depression generally have a lower abundance of beneficial microbes such as bifidobacterium and a higher presence of pathogenic bacteria like Proteus, which is known to cause cellular damage. This bacterial imbalance might be a significant pathological factor in IBS patients with depression. Additionally, functional pathways related to the production of short-chain fatty acids, SCFAs, which are typically reduced in depressed subjects, were found to be less prevalent in these patients, suggesting a potential biochemical link between gut health and mood disorders. The study also underscored the significant influence of demographic factors on IBS. Women and younger individuals displayed more significant microbiota compositional differences when compared to controls, indicating a higher susceptibility to IBS and more severe forms of gut dysbiosis. Age-specific trends in gut microbiota composition were noted, with younger people more susceptible to IBS-D and older individuals more susceptible to IBS-C. Diet was highlighted as a critical factor affecting gut microbiota across different IBS subtypes, which suggests that specific dietary interventions, like increased fruit intake or lactose avoidance, could alleviate symptoms in various IBS patient groups. This extensive study not only provides the largest deeply phenotyped IBS control cohort to date, but also intricately links gut microbiome profiles to depression, age, sex, and dietary influences, offering significant insights into the pathogenesis of IBS. Validation by an independent cohort reinforced the reliability of the findings, though the study acknowledges limitations, including potential confounders and biases related to data collection methods. Ultimately, these findings pave the way for personalized gut microbiome modulation strategies, aiming for the most effective therapeutic outcomes based on individual microbiome profiles. This approach could revolutionize treatment paradigms for IBS, potentially leading to more targeted and effective interventions based on each patient's specific microbial and functional profiles.